Let's talk about the first strategy for tissue replacement, organ repair via stem cell. Let me introduce stem cells to you. Stem cells have two remarkable qualities about them. Number one, they can divide many times. We have talked about cells that they cannot divide so many times because their telomeres get shortened every time. Stem cells have found a strategy to overcome this problem, so they can divide many more times than normal other body cells. Under certain physiological or experimental conditions, stem cells can be induced to become specialized cells. So stem cells, two very important properties. They can divide numerous times, more than other normal cells in the body, and they can be told to differentiate or become specialized cells of a particular tissue. There are two types of stem cells, embryonic and adult stem cells. We will talk about these two categories individually. First of all, let's talk about embryonic stem cells. They are derived from the embryos, and that's why there's an ethical dilemma, because when cells are taken from the embryos, as a result, the embryo dies. Some people equate that with murder, so that is why there's an ethical dilemma for using embryonic stem cells. However, the plus point of these embryonic stem cells is their ability, they have a greater ability to differentiate into pretty much any type of cell in the body. Also, they have unlimited ability to renew themselves. They can divide infinitely without having any consequences, whereas the ability of adult stem cells to divide is relatively limited. The other problem with embryonic stem cells is because they are foreign in origin, if you transplant these cells or organs made from these cells, these particular cells, the patient will have to be put on immunosuppressant because these cells are foreign and our immune system will react to a foreign entity in our body. Immunosuppressants, of course, they have their side effects. So the 2 points about these cells which are not favorable is that there is restriction on use of embryonic stem cells because of ethical dilemma. And the second problem associated with embryonic stem cells is that they would be foreign when they are transplanted, and they can cause immune rejection. So there are strategies that can be taken to overcome these two problems. First of all, let me tell you about the strategy that we can use to overcome the ethical dilemma, meaning a technique that will allow us to harvest cells from embryo without killing them. Secondly, how we can make sure that our embryonic stem cells do not cause immune rejection. Basically, we are going to change the genetic material present in these embryonic stem cells. So first, we are going to see an animation which shows blastomere biopsy, which is a procedure in which a single cell is removed from the embryo or the blastomere. And the beauty of this procedure is the embryo is still viable. So it overcomes the ethical dilemma. The second animation, we will see how we can change the genetic material of an embryonic cell. We will see that animation later on. First of all, I'll show you blastomere biopsy clip, how this procedure is done. So let's look at that. So here's an embryo. You can see it is at multicellular stage. The fertilized egg has divided several times. Here is the protective covering called zona pellucida. So we have to first remove the zona pellucida. This circle you are seeing is basically made by the targeting laser. This is where the laser will focus and punch a hole through this zona pellucida. So we are going to turn on the laser. Here you can see laser is going to make a hole. Here there is a hole here, and it is basically chewing away this protective covering. So now it will have enough room for this pipette to go in and pull a cell out. So we are almost done with the laser. So this part is now open, and we can use this pipette, glass pipette, specialized glass pipette, to go in and pull a cell out. So we will see that in a few minutes. So here's the cell. We are going to press this embryo, and now the pipette is going to go in and pull the cell out. I would just like to mention this is the holding pipette. It has a negative pressure, a suction pressure. Because of this pressure, this embryo is stuck to this pipette. Now we are going to use the other pipette to get a single cell out. it is going to go in, negative pressure will be applied, cell will be sucked into the transfer pipette. So there you go, the cell is squeezing, gently we are squeezing the cell through the pipette. Almost have it, almost. Okay, so the cell has been removed. The cell can be plated in a different dish, and we can get many more cells out of the single cell once it has divided. So we have solved one of the problems associated with use of embryonic stem cells, which is the ethical dilemma. So we have shown you a technique which can be used to overcome that dilemma. Next we are going to see how we can use a different technique, a similar one however, to overcome the second dilemma with the use of embryonic stem cells, which is that they can cause an immune rejection. But that would be in the next module.